Hello my dear students, welcome to KGH Academy. So in this video, in this session, I want to discuss a topic, paper 5, science and technology topic, cloning. So very important topic. So first of all, here I am giving cloning meaning, definition, types, etc. So cloning derived from a word is called clone. Clone means twig. A plant part is called twig. Plants propagated through twigs, branches, stem branches. This type of the propagation is called clonal propagation. Clonal propagated plants all are show same characters. For example, you see here, this is one plant, separated a branch, stem cutting, and here we are growing this branch in a pot, and this plant and this plant exactly same, like Xerox copies. Same this type of the method you can apply to the rose, jasmine, hibiscus, etc. Plants which are propagated through stem cuttings, they look like same, 100% same, Xerox copies. And this type of the propagation is called clonal propagation, and these are called clones. Same way, organisms, plants, insects, higher organisms, animals, clones look like same. Producing of same type of ones, same type of the organisms is called cloning. We can apply cloning to the normal material also. For example, credit card cloning. Credit card cloning means again same, producing of or developing of same type of another credit card with the help of our details. Like this, cloning means producing of same type of one, same type of the material, same type of the plant, same type of the animals. Here we are applying cloning to organisms or plants, animals, sometimes genes also. So producing of the same type of the genes, gene cloning. Producing of same type of the animal, animal cloning, like this. So here clone means twig. How? So plants propagated through twigs look like same way. Cloning animals, cloning plants, cloning organisms look like same characters. So in this way, so clone word derived or clone word formed. And next, types of cloning. So cloning basically two types: natural cloning, artificial cloning. Natural cloning means in nature, in ecosystems, in nature, cloning naturally present. Some organisms, plants, insects, bacteria, etc. So naturally contains, naturally shows cloning. They reproduce through the cloning. So this is called natural cloning. See here, natural cloning. Plants, I am already explained. Stem cuttings, clonal propagation. Through stem cuttings, clonal propagation, plant shows this cloning. And bacteria, algae, fungi, protozoa, all these all are microorganisms. Sometimes algae, fungi, large size of the organisms. These all are produced same type of ones, same type of organisms, identical ones, identical organisms through the reproduction, and this is called cloning. Naturally all these organisms, they continue the next generation with same type of the organisms, same type of individuals. So this way all these plants, bacteria, algae, fungi, protozoa shows natural cloning. So here we are discussing detailedly about the artificial cloning. Artificial cloning. One more slide related to animal clones. So very clear. See, natural clones in animals are rare than in plants, fungi, bacteria. Okay, so example, Hydra obelia will produce new organisms by budding. So budding is producing same type of the organism. That's why it's indirectly one type of the cloning. Aphids will produce large number of identical females offspring by parthenogenesis in summer. Parthenoid virgin genesis producing without fertilization organisms produces. So that is called the parthenogenesis. Without fertilization means they look same. That's why cloning in animals is called asexual reproduction. Cloning in animals, okay, in any animal, particularly higher animals, that is equal to asexual reproduction. Actually, asexual reproduction not present in higher animals. Asexual reproduction present only in lower animals. Asexual reproduction meaning without gametes, without production of gametes, without formation of gametes, and without fusion of gametes, without formation of zygotes, directly organisms develop. So this type of the reproduction is called asexual reproduction. Here in cloning, there is no formation of gametes, there is no fusion of gametes. Okay, so directly organisms develop. That's why cloning is the asexual reproduction. The lower animals shows asexual reproduction. That means they're producing new organisms next generation through cloning. Okay, see, cleavage of fertilized ovum to give two separate embryos will produce identical twins. So indirectly in humans, identical twins also indirectly one type of the cloning, natural cloning. Okay, nature, because of some reasons what happens in nature, so here embryo split into the two parts, that's why two identical twins are formed. This is also examples of the natural cloning. So without our interference here in nature, naturally, so clones are produced. That's why these are called natural clones or natural cloning. So next, artificial cloning. Again, artificial cloning, different types: gene cloning, therapeutic cloning, reproductive cloning. Producing of same number of genes, same number of gene copies, DNA copies, hundreds, thousands, lakhs is called the gene cloning. And cloning experiments because of the therapy treatment is called therapeutic cloning. Cloning process, cloning techniques for reproduction purpose, to continue next generation, producing of a new individual, new organism, new animal is called reproductive cloning. Again, we are discussing these three detailedly. So first, gene cloning. Gene is nothing but part of the DNA. Here we can generate the lakhs of DNA copies with the help of two methods. One is by using vectors, second one PCR mission. So vectors, first vector method, by using vector method or by inserting vector into the host, so we can produce the or we can generate the lakhs of our same gene copies. Here I am giving detailed analysis about this. By vector methods, how we can produce the gene copies or genes. Here vectors are nothing but plasmids. Plasmids we are using as a vector. What are the plasmids? Plasmids are extra DNA copies, circular DNA copies present in bacteria. So bacteria cell contains like this round or circular DNA pieces. Apart from this chromosome, so extra circular pieces called plasmids. So plasmids we are using as a vectors. First we are inserting our interested gene in this plasmid, and we again transferring into the host. When host multiply, automatically our gene multiply, or plasmid multiply, automatically our gene multiply. This way we are using a vector called plasmid. We are increasing, we are producing n number of copies. This is nothing but gene cloning. To understand this, here I am giving a slight gene cloning method. See, the first is to choose the appropriate DNA to clone of genomic DNA or cDNA. Okay, and next, collection of DNA fragments, cutting and vectors, insert the DNA fragment into the vector by using different enzymes, and introduce DNA fragments into population bacteria. Here this we are using as a host. Okay, vector means plasmid. Select the colonies containing desired sequence of from the library. To understand this, here I am giving one clear slide. And see, this is the eukaryotic chromosome. Eukaryotic chromosome means eukaryotic means organisms which contain real nucleus. They are called the eukaryotic chromosome. So chromosomes contains DNA. So we are isolated a DNA from this chromosome. See, this is isolated DNA. By cutting DNA with help of restriction enzymes, also called molecular scissors, we are isolated a DNA or gene. See, DNA fragments of by obtained by cleavage chromosomes, restriction endonucleases. So restriction endonucleases helpful to cut the DNA at particular site. That's why restriction endonuclease also called molecular scissors. With help of scissor, how we are cutting a material same way. This enzyme cut the DNA at particular point. Okay, so by using restriction endonucleases, here we are isolated a gene. Okay, from a organism or eukaryotic organism chromosome. And next, this site, we are isolated the plasmid, cloning vector. Here vector means which transfer from one place to another place. General meaning vector means it transfers something one place to another place. Here we are using plasmid as a vectors because plasmid transfer, so our gene, interested gene from one place to another place. So that's why we are taking plasmid as a vector. See here, cloning vector plasmid. And next we are cutting this plasmid with same restriction endonucleases. After cutting, next we are adding this, our interested gene into here and pasting it. Okay, pasting with ligase. See here, cutting of this plasmid here. And next we are adding this gene here. Next pasting, DNA fragment are ligated the prepared to cloning vector. Now, so our interested gene present in this vector. And next we are introducing this plasmid which contains our interested gene into the host. Here bacteria act like a host. By different methods, by using different method techniques, we are inserted, we are transferred this plasmid which contains our interested gene or DNA into the host cell. See here, plasmid with our interested gene. So this is inserted or transferred to a bacteria cell act like a host. When host multiply, automatically our interested gene multiply. See here, 1 bacteria, 2, 2, 3, 4, 3, 2, 4, 8, 16 like this. When bacteria multiply, automatically our interested gene multiply. So like this, with help of this gene cloning, we can create the hundreds, thousands of gene copies very easily. So this is the vector method or host method, vector host method. So apart from this, by using one special machine called PCR, polymerase chain reaction, also called thermocycler. So this is a mission. With help of this, we can amplify, we can produce a more number of DNA copies. Okay, so these are the few points in gene cloning. Next, very important, the real cloning is therapeutic cloning or reproductive cloning. Cloning experiments, particularly SCNT, somatic cell nuclear transfer, SCNT experiment for therapy treatment, that is called therapeutic cloning. SCNT experiments for reproduction or continue next generation to producing another animal is called reproductive cloning. These two, therapeutic cloning, reproductive cloning, totally depends upon a technique called SCNT. Very important. Gene cloning etc. is not based on this technique. So total cloning based on a technique is called SCNT. You can expect a separate question on this. What is a cloning experiment? And draw that schematic diagram of SCNT. Or cloning methodology. Explain about cloning methodology. Based on which experiment or by using which experiments we can produce the new organism, cloned organism like this. So these two are mainly based on a technique called SCNT. See, SCNT. SCNT meaning somatic cell nucleus transfer. This is a technique, experiment, okay, to produce clones. We are using this SCNT for two purposes, two ways. One is to isolate embryonic stem cells, and embryonic stem cells we are using for treatment, therapy. So this type of the cloning is called therapeutic cloning. Same SCNT experiments for continue next generation production of producing of new individual, new organism, that is called reproductive cloning. Here these two are cloning, producing of animals, organisms through cloning is equal to asexual reproduction. I am already explained. In asexual reproduction there is no gamete formation, fusion and fertilization. Without that organisms are developed. That's why this is called asexual reproduction. Here I am explaining about the somatic cell nucleus transfer. Somatic cell nucleus transfer experiment first conducted by a scientist called John Gurdon. He is called father of cloning. Okay, Ian Wilmette is not godfather of cloning. No doubt first complete higher animal, dolly, okay, sheep produced by Ian Wilmette. By using SCNT technique, he produced dolly. But first SCNT technique developed by John Gurdon. That's why John Gurdon is called godfather of cloning. Don't confuse between these two scientists. Ian Wilmette continued this John Gurdon experiment. John Gurdon first developed this SCNT experiment. By using SCNT experiment, Ian Wilmette produced dolly. So here cloning technique, SCNT technique first developed by John Gurdon, that's why he is godfather of cloning. So for this, he awarded Nobel Prize in Medicine 2012 along with Shinya Yamanaka, who are developed IPS, induced pluripotency stem cells along with him. So he awarded Nobel Prize in 2012 medicine. To understand this, here I am giving a slide. This is therapeutic cloning. Here what we are doing? We are isolating a body cell, somatic cell, body cell, okay, and separated this is the nucleus. From a female, we are taking a oocyte, means egg, and we are transferred this into here. This is nothing but SCNT, somatic cell nucleus transfer. Here we are transferred a somatic cell, body cell into a empty egg. So this is SCNT. And this embryo developed and blastocyst stage, 2 cell, 4 cell, 8 cell, 16 cell, 32 cell stage, embryo is called blastocyst. From blastocyst stage, we are isolated embryonic stem cells. And by using embryonic stem cells, here we are developed some muscle cells, etc., or other type of the cell, and again introduced in person body to treat different types of diseases for treatment purpose. So this way, somatic cell nucleus transfer technique for therapy or to isolate embryonic stem cells, this type of the cloning is called therapeutic cloning. Okay, again to understand this, one more slide. It's a very clear. See, somatic cell. Somatic cell. Somatic cell nucleus red color. This one. Okay. And here egg cell, oocyte. Here they are removed this. Okay, egg from here, somatic cell. Once again, this is egg cell, they are removed nucleus. And this is somatic cell, they are removed nucleus. And they transferred this somatic cell nucleus into egg cell. This cell. They removed this one. They separated this nucleus. Oocyte means egg. From female, we are isolating a egg or oocyte and removed nucleus. Next, we are isolated a somatic cell, body cell from a person, male or female. Okay, and same isolated nucleus. Next stage, we are transferred this isolated nucleus from somatic cell transferred to the nucleus. So that's why this is called SCNT, somatic cell nucleus transfer. After here, we are utilizing this embryo, blastocyst embryo, in two ways. In case this is introduced into the surrogate mother uterus to develop, to produce a baby, is called reproductive cloning. In case we are using somatic, this embryo to isolate embryonic stem cells. Again, we are using embryonic stem cells for treatment in person to different disorders. And this process is called therapeutic cloning. So this is therapeutic cloning and this is reproductive cloning. Up to here, method same. That means through somatic cell nucleus transfer, producing of an embryo, up to here, same. From here, how we are utilizing this embryo? Based on that, here cloning two types. Utilizing of this embryo to produce another organism or another animal, same type the animal. Okay, this process is called reproductive cloning. Here another organism reproducing it. Same embryo used to isolate embryonic stem cells to treat different type of the diseases. This process is called therapeutic cloning. So this slide is very clear. Somatic cell nucleus transfer and therapeutic cloning, reproductive cloning. Next slide. This is a John Goodon experiment. Somatic cell nucleus transfer experiments. He taken one egg and isolated nucleus, and they are taken frog tadpole larva body cell and separated this nucleus, and he transferred this nucleus into this egg. This egg contains somatic cell nucleus, and this process is called somatic cell nucleus transfer. After this become embryo, embryo become tadpole larva to frog. Okay, same these techniques, next scientist, they are applied to the animals. This way, first somatic cell nucleus transfer conducted by, and first cloned animals by using SCNT conducted by John Goodon. John Goodon produced a frog or frog tadpole larva by using SCNT technique. That's why it's called godfather of cloning. And next, same frog egg, unfertilized egg. Okay, so they remove nucleus, and next frog body cell nucleus they remove. See, this is a body cell nucleus. With help of a micropipette, they introduce this body cell nucleus into the egg, and now this becomes zygote converted to embryo. So embryo to next frog tadpole larva to frog. Okay. This way, by using SCNT technique, John Goodall developed a frog tadpole larva or frog. Next, same. Here you see somatic donor. Somatic donor means nucleus donor. Okay. Here somatic cell nucleus donor. And next, egg donor. Okay, a cow. So here they taken this egg with nucleus. Then they are separated this nucleus. And somatic cell nucleus separated. This is very clear. This is yellow color, this is a black color. And they are transferred this nucleus into egg. And they are multiplied and next introduced into the same this egg donor, surrogate animal. The produced one like this. Here cloned animal always look like same like nucleus donor, not egg donor. Normal words we can say is like this. Cloning animals always like donor, because donor nucleus contains chromosomes, chromosomes contains DNA, DNA nothing but genes. So that's why same like donor, egg donor, somatic cell nucleus donor. Here somatic cell nucleus donor, this animal. That's why cloned animal always like this. Not only this animal, all animals, all higher animals, clone animal same like donor. Donor means somatic cell nucleus donor. Next, again same. Okay, so here egg cell, nucleus separated, body cell, nucleus separated. Okay, this is empty egg cell. Here I am giving 3 to 4 different slides to understand the topic. Through picturization form, I think you can understand very easily. That's why I am giving. So I am explaining same information with help of 2 to 3 different type of slides. See here, okay, they transfer this into the here and cloned cells. This cloned cell surrogate mother, that is reproductive cloning. So, and isolating the stem cells, that is called therapeutic cloning.